Stop doom scrolling for 5 minutes and learn this MCAT topic.
In this video, we're going to talk about the innate immune cells. Now, broadly, the innate immune cells can be divided into granulocytes and agranulocytes. Now, why are they called this? Well, granules, that's where the word comes from. Granules are these little sacs of proteins that are found in the cytoplasms of some of these cells. And if you stain some of these cells and you look under a microscope, if you see the granules inside the cytoplasms of the cell, those are granulocytes. And the first granulocyte is going to be a neutrophil. Now, these are the most abundant immune cells in the bloodstream. And these are sort of the first responders of the immune system. They're kind of like the EMS of the immune system because if there's an inflammation or an infection somewhere, the first white blood cells that get there are the neutrophils. Now, also these are phagocytic, so they kind of eat pathogens like bacteria. And actually, when they eat a bacteria, for example, the granules inside these neutrophils will open up and release proteins that can help break down that bacteria. Now, these are also very short-lived, and these also, like the debris from the neutrophils when they die, forms pus. Now, the next group of innate immune cells and granulocytes are the basophils and the mast cells. And I put these two together because they are very similar, but where they are found differ. So basophils mostly circulate within the bloodstream, whereas mast cells are actually found mostly inside the tissue. They don't really circulate in the bloodstream. Now, both of these release histamine. That's what I want you to remember. They release the molecule histamine, which is derived from the amino acid histidine. And these really mediate allergic reactions. Now, the next cell is the eosinophil. This is the last granulocyte. Now, eosinophils actually fight parasitic infections. That's what I want you to remember. And these are also implicated in asthma and also autoimmune diseases. But really, for the purposes of the MCAT, remember eosinophils are related to parasites. And next, we have the agranulocytes. So for these cells, if you look under a microscope, you will not see any granules in their cytoplasms. Now, the first group, this is really important, are called monocytes. Now, this can be a little bit confusing, but the idea here is that monocytes are the form that actually circulate in the bloodstream. And when they enter a tissue to fight, say, an infection in that tissue, they can actually differentiate or evolve, so to speak, into two different types of cells. The first are the macrophages, right? These I would remember as being the fighters. So these are phagocytic, kind of like neutrophils, but unlike neutrophils, they are not short-lived, they're long-lived, okay? And these are also antigen-presenting cells, meaning that they express major histocompatibility complex 2 and can actually present antigens and activate helper T cells, okay? However, what you should remember is that the macrophage form, its primary function is to fight pathogens, not to present antigens, even though it can do that. The other type of cell monocytes can form are the dendritic cells, and these are the scouts and recruiters. So these are also phagocytic, and they can also destroy pathogens. However, just like macrophages, they express MHC2, and they can present antigens. And it's the dendritic cells whose primary role is actually to present the antigen to the helper T cell, the CD4 cell, and activate it and recruit it to the site of infection. And then the last innate immune cell and the last agranulocyte we're going to talk about is the natural killer cell. Now, these, what I want you to remember, these are antiviral and anti-tumor cells. And what they do is they kind of circulate in the blood and they can enter tissues and they can detect cells that are possibly infected with the virus or possibly tumor cells. And when they do so, they release these molecules called perforin and granzyme. And the idea is that perforin will actually create like an opening in the cell, and then granzyme will enter the cell and induce apoptosis in those cells. And also these cells release a cytokine, a signaling molecule called interferon gamma or INF gamma, which helps to activate macrophages.